Hello, everybody. Welcome to Ms. McGuire online video lecture. And today we're covering human genetic chromosomes. This video chapter will be divided into two parts. In first part, we will cover structure of chromosomes, and in the second part, we will talk about chromosomal abnormalities. So let's go ahead and begin. We're going to start with learning outcome. After studying this chapter, you will be able to list the major parts of a chromosome, list the types of chromosomes based on centromere position, describe the way that chromosomes are obtained, prepared, detected, and depicted in detail, explain how atypical chromosome numbers arise, distinguish polyploidy from aneuploidy, describe specific aneuploidy conditions, distinguish between the consequences of having balanced versus unbalanced chromosomes, describe deletions, duplications, and the two major types of translocation, distinguish the two types of inversions, describe the consequence of having an isochromosome, describe the consequence of having a ring chromosome, explain how a person could inherit both copies of a DNA sequence from one parent, and describe how inheriting both copies of DNA from one parent can affect a person's health. So, cytogenetics is the classical area of genetics that links chromosome variations to specific traits, including illnesses. Excess genetic material has milder effects on health than a deficit. So what it tells you that, well, we're supposed to have precise number of chromosomes, right? So humans have 23 pairs of chromosomes. You get 23 from one parent and 23 from another parent. But sometimes we can get more than 23 pairs. 23 pairs is 46. So sometimes we might have more than 46 chromosomes. And sometimes, I'll take it back. We cannot have it because we already have the number of chromosomes that always be with us, right? So you are born and you have your chromosome numbers, and most likely you have 46 chromosomes. Just some kids, some people are born with less than 46 or more than 46 chromosomes. And that's what it says. If we have more genetic material, then it has mild effect. But if we have less, then we have more severe defect. So large-scale chromosomal abnormalities present in all cells disrupt or halt prenatal development. As a result, only 0.65% of all newborns have chromosomal abnormalities and produce symptoms. That means, how many newborns have these chromosomal abnormalities? How many newborns have symptoms that are related to chromosomal abnormalities? For example, they have extra chromosome, or they're missing a chromosome, or they're missing a part of a chromosome. How common is that? It's actually only 0.65%. Why? Because usually when fetus has abnormalities, it will not develop, right? So prenatal development would stop, and pregnancy, unfortunately, will result in spontaneous abortion. So that's why we don't have like huge percentage of newborn with chromosomal abnormalities, because it's really very severe condition. Chromosome consists primarily of DNA and proteins with a small amount of RNA. So if you look at a chromosome, mostly it's made of DNA, but it's also made of proteins called histones, and DNA is wrapped around those histones, and it has small amount of RNA. Now, chromosomes are distinguished by size and shape. That means all your 46 chromosomes are not the same. Some are longer, some are shorter, some have a, and they might have different shape. Now, the essential part of a chromosome's essential parts are telomeres, origins of replication sites, and centromere, right? So all chromosomes will have the three parts. So let's look at the diagram of a chromosome. In this situation, right in the middle here, you have centromere and you have arms. Now, for this particular chromosome, we have a shorter arm and a longer arm. The shorter arm is called P arm and longer called Q. Right now, chromosomes are made of this chromatin, right? The chromatin is made of DNA, proteins, and a small amount of RNA. Now, the end of chromosomes, this part is called telomeres, right? So you have telomeres on the both ends. In the center, we have centromere, and we have different type of chromatin. We have heterochromatin that is colored in darker color. When we stain a chromosome, it appears dark. And we have euchromatin that's stained lighter. Now, if you look over here and you see this X shape of a chromosome, this is one chromosome that was already replicated. So it's kind of like if you look at the middle, so that's a one chromosome and it's made identical copy, well, as identical as it can be, because every time we make copy of DNA, we have small mutations. It happens always, but they're supposed to be identical. So here's the chromosome, and this is its copy. And these two parts of one chromosome are called sister chromatids. Now let's look what this heterochromatin is and how it is different from euchromatin. Now, heterochromatin is darkly stained, and it consists mostly of repetitive DNA, the DNA that does not code for proteins. Euchromatin is lighter stained, and it contains most protein encoding genes. Now, telomeres are those end parts of chromosomes. Those are tips of chromosomes. They composed of many repeats of TTA, GGG. So those repeats are actually, they do not code for any protein. So those are not genes. You don't have genes in the telomeres. Telomeres just exist to prevent your genes from damaging. So with every division, when you divide, when your chromosomes divide, those telomeres get shorter and shorter. And they believe it contributes in a aging process of our cells. Now we also have, remember, centromeres. So largest constriction of the chromosome and where spindle fibers are attached. So where chromosome is constricted, that's called centromere. You know, it's interesting that centromere doesn't necessarily need to be in the center of chromosome. We're gonna look at it in a minute. But when your cell goes through a process of division, when cell divides, then you need to separate those sister chromatids. And to do that, you need to attach the special proteins called a spindle to a chromosome and pull them apart. And where spindle is attached to chromosome, that's called centromere. So it's lie within vast stretches of heterochromatin. Bases that form the centromere are repeats of a 171 base DNA sequence. So again, we have repeats of 171 base sequence. Centromeres, this part are replicated at the end of S phase and include a special protein called centromere protein A, or CANP, centromere protein A. And it's passed from one generation to next generation. That's for centromere. Then we have subtelomeres. Those are chromosome regions between the centromere and telomere, consist of 8 to 300 bases. Near telomere, the repeats are similar to telomere sequence and contains at least 500 protein encoding genes. About 50% are multi-gene families that includes pseudogenes. So now let's look at everything that we just described. I remember so that's a chromosome. It's only show us a part of it, so only one arm. So here's a part of another arm, and here's our most constricted part of the chromosomes that's called centromere. Right then, we have our at the end. Remember, we have maybe right there, yeah, right there. Sorry. So that's we have a telomere. The end, right? In the center, we have centromere. Now, telomere is a repeat of TTAGGG. So you see it's shown here, TTAGGG, TTAGGG, and you have TTAGGG, and so on and so on. And with each division, this telomere gets shorter. Right? Under the telomere, you have subtelomere, right? So that's your subtelomere. That's located between centromere and telomere. And this is where you have, so you will have genes that code for proteins. But before we get to this part, let's go back to look at the chromosome again. You see how we have this heterochromatin, right? So heterochromatin here and heterochromatin over here. So this part doesn't code for protein. So where we have this lighter parts, chromatin, this is where we have protein coding genes, right? So that's why it shows over here, we have 6 base repeats similar to telomeres. This is when you get close to the telomere, you get closer to telomere and repeats became similar to telomere. And then you have a repeats, we call them short repeats, that code for most of the genes. Now, let me just go back here for a second to read about, kind of return back to subtelomeres. So it's a chromosome region between the centromere and telomere, right? So if you look over here, right, between the centromere and telomere, and it's made of from 8 to 300 bases, and that contains at least 500 proteins. And where it's located, subtelomere, is over here where we have this heterochromatin. And that's mostly where you have those genes that code for protein. And remember, the whole purpose of your DNA is to code for protein. Now, what is karyotype? It's a chromosome chart. It's a major clinical tool. Displays chromosomes arranged by size and structure. Humans have 24 chromosome types. Autosomes are numbered 1 through 22. So we have chromosome 1, chromosome 2, chromosome 3, so on, up to 22. And we have sex chromosomes, either X chromosome or Y chromosome. So here the karyotype, so this is arranged picture of human chromosomes, and they divided into groups, group A, group B, group C, D, E, F, and G, and here's our sex chromosomes. And you can see that here's a chromosome 1, so they go from larger chromosome to a smaller chromosome. So chromosome 22 is the smallest one, and we, of course, not including the sex chromosomes. So we arrange them from longest, from biggest to smallest. So we have 2 chromosomes 1, because you get chromosome 1 from mom and chromosome 1 from dad. You have 2 chromosomes 2, one from mom, one from dad, 2 chromosomes 3, right, and so on, 2 chromosomes 4, 2 chromosomes 5, right, and so on. So you have 22 pairs of autosomes and 1 pair of sex chromosome. Now, if you have X and X, you're female. If you have X and Y, you are male. But if you look at this karyotype, you see how we have extra chromosome 21. So it's abnormal karyotype. This is karyotype of a person with Down syndrome, because Down syndrome, another name for it is trisomy 21. When we have 1 extra chromosome 21, that result into some changes in a phenotype of a person. Right, so that's what karyotype is, right? So we arrange chromosomes in a order and we give them numbers. So karyotypes are typically performed on cells during mitotic metaphase. So we need to extract DNA from a cell, but when exactly you want to do it? You want it when cells start dividing, right? Then DNA coils tightly and you can really extract nice chromosomes without damaging them. And it's also make it easy to visualize. Karyotypes are useful at several levels. They can confirm a clinical diagnosis, reveal effects of environmental toxins, and clarify evolutionary relationship. Now here we said that we have different type of chromosome. It can be telocentric, when the centromere is at the tip of the chromosome. So here's telocentric, so here's our chromosome, and here's our centromere. Acrocentric, close to the center, right? So right there, we have acrocentric. Submetacentric, it's off center, and metacentric at the center. So that's a different type of chromosome based on location of centromere. Actually, acrocentric cannot be close to center. It's close to the tip of chromosome. Yes, I think this is just the typo over here. So we have telocentric, acrocentric, close to the tip, submetacentric, and metacentric. Chromosome can be imaged from any type of human cell that has a nucleus. If cell doesn't have nucleus, of course, you cannot see chromosomes and perform karyotype analysis. For example, RBCs or red blood cells, they do not have nucleus. The most common application of human chromosome testing is in prenatal diagnosis. Chromosomes are also checked in relatives of people known to have atypical chromosomes to explain infertility or to diagnose or track cancers. When do we need to look at human karyotype? And remember, karyotype is this picture of chromosomes. So when do you need it? Like, have you ever done your karyotype taped? Right, so probably nobody never looked at your karyotype. Nobody looked at my karyotype, that's for sure. So when do we need to have this karyotype testing done? In prenatal diagnosis, when we have maybe probability that a child will carry some genetic disorder, right? Or if we know that we have people with abnormal or atypical number of chromosomes or type of chromosomes, or for example, we have a couple and they cannot have kids, so we want to look at their chromosomes and see if everything is normal, or it might be also to track some cancer. Now, how do we extract the chromosomes for testing? There is older approaches called amniocentesis and chorionic villus sampling, or CVS, and new approaches that called cell-free fetal DNA testing. So let's look at these techniques. So the first one is amniocentesis. A needle is used to remove a small sample of amniotic fluid from the uterus of a pregnant woman, and ultrasound is used to follow needle's movement. So you take, when baby are inside the uterus, it's surrounded by amniotic fluid. So there is a fluid over here. It's called amniotic fluid. So we just put a needle with a syringe and just draw some sample of this fluid, right? And that will be DNA in this fluid. So we can do karyotype or other analysis that we need. It's performed at fetus of 15 to 16 weeks old. Right, this is our amniocentic technique. The next one is chorionic villus sampling. This can be performed a little bit earlier in pregnancy, 10 to 12 week, provides earlier results than amniocentic, but does not detect metabolic problems and has a great risk of spontaneous abortion. This way, in this technique, we collect fluid from a chorion. And chorion is a part of the fetus and mother's uterus that form placenta. So you pretty much draw the fluid, and placenta is rich in the blood supply. So we just draw a little bit fluid from a placenta and analyze DNA in the placenta. So as you see, this involves some needles that you need to insert into uterus, and you have fetus that is developing. So you can expect some complications from this method. That's why for many years amniocentesis was limited to women over age 35, because we don't want to put baby in risk if mother is still young, because when mother is older, the frequency of genetic disorders increases rapidly. So over here you can see maternal age in years and frequency of for example, trisomy 21, that is Down syndrome in live-born infants, and you see how rapidly the chances to have baby with Down syndrome increases when mom gets older. But when mom is young, the chances are very low. That's why they performed this amniocentesis only on women over 35, because the risk of the procedure equals the risk of miscarriage, which rises with maternal age, right? But okay, let me see if I missed something. Yeah, so let's cover this slide first. But today we use a new technique called cell-free fetal DNA testing, and that's why we can. It doesn't have a risk of complication or termination of pregnancy. That's why we can test even younger mothers. So in a pregnant woman, about 20% of small DNA pieces found in the blood that come from the placenta, and therefore represent fetus. So if we have pregnant female, in her blood, we will find DNA that comes from placenta, and that DNA represents the fetus. Tests of cell-free fetal DNA are done at 10 weeks or later and are rapidly replacing older techniques called non-invasive prenatal diagnosis or testing. Analysis of cell-free fetal DNA is based on proportions, and entire genome can be reconstructed, baby genome can be reconstructed from the DNA pieces. And so here's the next slide, shows us that this is mother blood inside the blood vessels, right? And you have a plasma, white blood cells, red blood cells, and you have some DNAs over here. They're free DNA, cell-free, so they're not inside the cells, they're just in the plasma. You have maternal cell-free DNA and fetal cell-free DNA that came from that placenta. And because the fetal cell-free DNA are very smaller fragments, we can differentiate between mom DNA and baby DNA, and we can pretty much reconstruct the whole genome of a baby, right? So that's a easy cell-free fetal DNA testing. Now, what are we talking here? We're talking here that, well, if we believe that a baby might have some genetic disorder, right? For example, Down syndrome, we can constrict the karyotype. So for example, this karyotype shows us we have one extra chromosome, so that's Down syndrome. Now, but how to obtain this DNA from baby who is not born yet, right? So that's why we can use several techniques, right? Is the amniocentric and chorionic villus sampling, or CVS, there are older approaches and they put baby into risk. Or we can use new approaches when we just draw blood from a mother, and in mother blood we will find a cell-free DNA of the fetus. And then we can make karyotype and we can look at the chromosomes, are they okay or not, right? All right. Now, when we obtain those DNA, so we get DNA from a DNA of a fetus, we're gonna stain it. So early karyotypes used generalized stains. These could not distinguish chromosomes in similar size, which were considered in groups. So the early karyotype was just represented by groups. You see how here we have group A, B, C, D, E, F, and G, because they all similar size. So we just say like, okay, well, one group, let's say group G, instead of 4 chromosomes, we have 5. So it means most likely it's extra chromosome 21, or it might be extra chromosome 22, because we were not able to distinguish between chromosomes of the similar size, right? Now we have improved staining techniques that give banding a pattern that unique to each chromosome. So now how we dye chromosomes, they have different bands, dark band, light band, dark, light, and it's unique to every chromosome. So it's easy for us to identify, is it chromosome 21 or 22? So the technique called FISH, or fluorescence in situ hybridization, when DNA probes labeled with fluorescent dye that bind complementary to DNA sequences on the chromosomes, use an algorithm applied a unique false color to each chromosome, painting and karyotype from several fluorescent dyes. So what it says, we can use, so on the top it says like, okay, before we use like a general stain and we just divided chromosomes into groups. Now we can use improved technique when give chromosomes banding appearance. We also can use FISH technique. And what FISH technique does, we have fluorescent dye. And this fluorescent dye attaches to a specific chromosome. And it allows us to see if we have any abnormalities. So for example, look at the next slide. So over here, we have our 46 chromosomes, but we use specific dye that attaches only to chromosomes, a very specific part of chromosome 21. And when you use this fluorescent dye, now look, you see 1, 2, 3 chromosomes picked up or made connection with this dye. That means we have 3 chromosome 21. That's what this fish technique is. Okay, so now we already discussed this cell-free fetal DNA testing, when you pretty much just draw a blood from a mother and you can reconstruct fetal genome. Okay, now if we can extract chromosomes from a cell, and we will dye it, right? And we will have different bands. Then it's easy for us to kind of like each band can have a name, and it allows us to put like a map on a chromosome. So imagine you have a chromosome and you have specific location. Like how would I tell you now this gene for example, for cystic fibrosis is located where exactly on a chromosome? How to explain? So the chromosome is divided into different sections. So that's called, this process called short band. So information in a karyotype is abbreviated by listing the chromosome number, sex chromosome makeup, and atypical chromosomes. So I'll show you how it looks, and then we get back over here. So for example, we did karyotype over person, and it's a normal male. We need to abbreviate it somehow, right? So we abbreviated 46. That means it has normal number 46 chromosome, XY, and XY tell us that's a normal male. Now this one, 46XX, that is a normal female. Now look at this, 45X. That means this person has only 45 chromosomes instead of 46. And what is missing? One X chromosome is missing, right? So a female with one X chromosome only instead of two X chromosome. Now this one, 47XXY. This person has 47 chromosome instead of 46 and one extra chromosome. And in this situation, when we have XXY, it's extra X chromosome because that's going to be a male with extra X chromosome. Now this, look at this abbreviation of karyotype. 47, one extra chromosome, XYY, so one Y chromosome extra. So it's a male with extra Y chromosome. Now look at this one. That's a really beautiful. So let's look. 46, that means normal number of chromosomes. XY, that's a male. But now look over here. It's a deletion of chromosome 7 of the short arm of chromosome 7. So del stands for deletion. So we have one chromosome that lost part of it. So chromosome 7, and remember how we have a short arm and long arm? Long arm is called Q arm. What did I say? Short Q, I'm sorry, it's a long because P before Q, so short before long. So we have deletion. Let me draw so it doesn't disappear. So here's chromosome 7, short arm and long arm. A P is short, Q is long, and part of a chromosome is missing. So it's a deletion on a chromosome 7, and it's in the long arm. Now let's look at this. One extra chromosome, 47, right? It's a female and has extra chromosome 21. So that's our Down syndrome, trisomy 21. Now look at this one, 46, normal number of chromosomes. It's a male. And now look, T, translocation between chromosome 7 and 9. That means a piece of chromosome 7 was attached to chromosome 9 and a piece of chromosome 9 attached to chromosome 7. So it's a chromosome that messed up. Chromosome 7 has a little part of chromosome 9 and chromosome 9 has a little part of chromosome 7. That's what translocation is. But where exactly this translocation happened? It happened at a short arm of chromosome 7, and specifically a location is band 21.1. And the long arm of the chromosome 9, and specifically at a band of 34.1. So it's a male with a translocation between the short arm of chromosome 7 at band 21.1 and a long arm of chromosome 9 at band 34.1. And yeah, they give these numbers to the band. We're not going into detail, but when we have this abbreviation, it just tells you exactly what's happening with karyotype of this person. Now look at this one. What do you think? It's 48 chromosomes. What do you think? Is it male or female? If you have Y chromosome, we're always gonna have male, but this male that have extra Y and extra X chromosome. Yeah, that happens too sometimes. So this is the abbreviation. Now remember, I told you we will go back. And that's what it says, that information in a karyotype is abbreviated by listing the chromosome number, sex chromosome makeup, and any atypical abnormalities. Symbols and shorthand describe the type of aberration. After the shorthand, numbers corresponding to bands and subbands. So remember how we had these numbers, like translocation, where exactly it happened. Chromosomally normal male is designated 46XY and female 46XX. Bands and subbands identify specific genes. So where exactly genes are located. Okay. All right, so here's our ideogram. It's a graphical representation called ideogram. Depict the arms P or Q, bands, subbands of chromosome aberration. Major regions are delineated by banding patterns. So here's our chromosome with the constricted part called centromere. Short arm is a P arm, long is Q, and then you see those bands. So we use specific technique that color different parts of chromosomes into darker or lighter bands. So that's a band, and this is subband, and we give them numbers, right? So, so for example, over here, it tells us that is a band 21.1. So it's a band 21 and subband 1. Oh my goodness, right? Okay, so that's where we're gonna stop this lecture, because the rest of PowerPoint covers chromosomal abnormalities, right? So thank you very much for watching, and I hope it was helpful.